Well, hello everyone and welcome to our last video on the topic of transport in plants. I wonder what events have transpired since our last video. Well, we were talking about transpiration and we focused on the transpiration pool. In this video, we're going to talk about transpiration again, but we're going to focus on transpiration itself. So we're going to talk about why is transpiration important, how is it studied through different experiments, what factors affect the rate of transpiration, and also this thing about wilting. Okay, so here are the textbook pages for your reference. Now you should recall that transpiration is the loss of water vapor from the aerial parts of the plant, mostly through the leaves and mostly through the stomata. Now one question that might come to your mind is, why do plants transpire anyway? I mean, why waste that water? Water is such a precious resource, right? Water bills are going up. So for plants as well, right? Why waste that water? Why lose the water vapor? What's the point of transpiration? So why transpiration? Well, it turns out plants just can't help it. You see, transpiration is an unavoidable consequence of gaseous exchange. Gaseous exchange here refers to the exchange of gases between the plant and its environment. And you should remember that plants do this by opening up their stomata. So they open up their stomata to exchange gases with the environment. Do you remember what that is for? Why do plants need to exchange gases with their environment? Well, hopefully you remember that, for example, for photosynthesis, plants need to take in carbon dioxide and they need to get rid of oxygen, which is produced in photosynthesis. So plants open their stomata up for gaseous exchange for photosynthesis, usually in the daytime. Now as a result of that, because the stomata are open, transpiration takes place, right? The stomata are open and so water vapor just naturally diffuses out of the leaf. It's unavoidable. Well, a simple example or analogy would be this. Suppose one day you were sitting inside your room and the windows are shut, okay, they're closed. And suddenly you smell something quite fishy, very smelly. Don't know where it comes from, maybe, let's not try to guess. But whatever it is, it's so smelly that you want to open up the windows to exchange the gases with the environment, right? So that the smelly gases can get out of your room. So you open the windows for ventilation. However, by doing that, other things can now escape, right? For example, if your room is air conditioned, then the cold air will also move out. Or if you kept a pet bird, the pet bird can fly out. So in the same way, plants open up their stomata for gaseous exchange, right? To exchange gases. However, the good stuff, for example, water, can also then be lost by transpiration, right? Water vapor will just diffuse out along its concentration gradient. So why transpiration? Well, the first thing is, it's an unavoidable consequence of gaseous exchange. Well, there are other reasons why plants transpire. There are other reasons why transpiration is important. But I would like you to read your textbook for yourself to discover what that is. Of course, one thing that comes immediately to your mind would be that transpiration is needed for transpiration pool. Right? You need it so that you can actually transport water and mineral ions up the plant. Now go read your textbook to find out the rest. Alright, first we're going to move on to the question of how do we study transpiration in plants? What experiments can be conducted to study transpiration? Right, after all, you cannot see the water vapor moving with your own naked eye. So we're going to talk about 3 different experiments. Okay, so on this page, you'll see the first experiment that can be used to study transpiration. And what you see here is a spring balance with 2 different setups. A spring balance, hanging on the spring balance is a plant, a twig, right, immersed in some water with a layer of oil on top of the water. Now I would like you to spend a moment to think about this experiment for a while. Pause the video here and have a think about these 3 questions. Okay, so how does this apparatus measure transpiration? Maybe you are thinking when something like this. Over here, shoot X has leaves. So with leaves, there will be transpiration, right? Water vapor will be lost from the leaves of the plant. Now as water vapor is lost, then water is pulled up the xylem of the stem by transpiration pull. Right? It's a suction force that pulls water up. So water will be pulled up from this test tube, up the stem, into the leaf, and then lost as water vapor. So over time, the amount of water here will decrease. And over time, the mass will decrease. So maybe from 30 grams, it will decrease to about 24 grams. So there's a loss of about 6 grams. And then we make the assumption, we assume that the loss of mass is due to transpiration. And from there, we can measure the rate of transpiration. In this case, maybe 6 grams of mass was lost in an hour. So the rate of transpiration will be 6 grams per hour. Now what is the layer of oil for? Now the oil here, the oil prevents evaporation of water. Because if the oil wasn't there, then water can just evaporate and there will be a loss of mass which is not due to transpiration. A loss of mass which is due to evaporation. So you don't want that. We want any loss of mass to be due to transpiration only. So we put the layer of oil there. Now finally, what results do you predict? So hopefully you predicted that the transpiration rate would be greater in shoot X. Because shoot X has leaves. And we learned that the majority of transpiration happens through the stomata of the leaves. Okay, so let's move on to the second experiment. Now what we see here is a plant in a plant pot, which is wrapped around with a plastic polythene bag. And it is placed on a weighing scale or weighing balance so that its weight can be measured. Now again, I want you to pause the video for a while and think about how this experiment works. Alright, so how does this experiment work? How can it be used to measure the rate of transpiration? Maybe you're thinking yourself again that there's a plant, there's some leaves, so there's loss of water vapor from the leaves, right, through transpiration. And that leads to water being pulled up the stem by transpiration pull. So water is pulled up from the soil and then eventually it is lost through transpiration. So over time, the amount of water from the soil will decrease. And therefore, the weight of this entire plant and the pot and the soil will decrease over time. So maybe we began with about 250 grams, right? The plant and the pot and everything. And after an hour, after 1 hour, we find that the mass drops to 240 grams. So there's a loss of mass of 10 grams. Now again, we assume that the loss of mass is due to transpiration alone. So from here, we can calculate the rate of transpiration. If in 1 hour, 10 grams of water vapor was lost through transpiration, then the rate of transpiration would be 10 grams per hour. Alright, because in 1 hour, we lost 10 grams of water. Now then, what is the bag for? Well, the bag wrapped around the soil, again, it plays a similar role to the layer of oil. It prevents evaporation of water. Okay, you can imagine if the bag wasn't there, then water can just leave the soil by evaporation, and that would result in a loss of mass not due to transpiration. However, this experiment only works if the loss of mass is due to transpiration alone. Right, let's move on to our final experiment, and in this final experiment, we have a device called the Potometer. Right, the Potometer. Right, so how does this Potometer work? Well, here we have our plant, a leafy twig. Okay, it's placed in the Potometer in the device, and this tube, this water tube, leads to a capillary tube, and into a beaker containing water. Right, so as with the previous experiments, we know that with transpiration there will be a loss of water vapor from the leaves of the plant. So water will be pulled up the stem of the plant by transpiration pull. Right, so water will be pulled up from this, it will be pulled up from the capillary tube, and actually ultimately pulled up from the beaker containing water. So the important thing to note is this: there is an air bubble in the capillary tube. So as the water vapor is lost, as transpiration takes place, this air bubble will actually also be sucked along the capillary tube. It will move along the capillary tube from here in this direction. So with these markers, we can actually measure the change in the position of the air bubble. Suppose the air bubble starts at position, let's just say 0 cm cube, alright? And it moves. So after a while, it moves to this point here, which is, let's say, 10 cm cube after 1 hour. So what we can say is that the rate of transpiration is 10 cm cube per hour, right? Because the bubble has moved 10 cm cube after 1 hour of transpiration. Now, what is this tab for? Okay, the tab just plays a role of resetting the potometer. It helps to reset the potometer. Because once the air bubble moves, let's say it moves very far to this point, we want to push the air bubble back to its original position. We just open the tab. And then we can reuse this potometer again. So I want you to take a moment to read your textbook and to think about this question. What assumption is being made when the potometer is being used? Okay, what assumption is being made? And a hint is here. Does the rate of absorption of water, that means the rate at which water is being absorbed into the plant, does it equal the rate of transpiration? Right, so read your textbook and come back in a short while. Now hopefully you've read the textbook and had a few thoughts, and you realized that the assumption that we're making is this: that the rate of transpiration equals to the rate of water being absorbed. Or if I want to put it differently, we are assuming that any water that is absorbed is due to transpiration alone. That means when this plant absorbs water, when this air bubble moves, it is only due to transpiration. However, is that a valid assumption? Does the rate of absorption of water really equal to the rate of transpiration? And the answer is not really. Why? Because the water that's absorbed here may not only end up being lost by transpiration. The water being absorbed here could also be used for something else. Now, what does the plant use water for? Right, the plant uses water for photosynthesis. So water is also used for photosynthesis. Right, remember how we talked about water being split during photosynthesis. Right, so water is also being used for photosynthesis. So some of the water that's being absorbed here is actually being used for photosynthesis. It remains inside the plant. It doesn't all get lost by transpiration. So this assumption is not fully accurate. The air bubble moves, the water is sucked up, not only because of transpiration, but also because water is used and required for photosynthesis. So we're not just measuring the rate of transpiration. In a sense, we're measuring a mix of the rate of transpiration and the rate of photosynthesis. Alright, but most of the time when we use the potometer, we assume that all the water being absorbed is due to transpiration alone. Alright, so I'll see you in the next video when we finish up talking about transpiration.